Okay, let's unpack this. Think about your morning cereal, maybe that sandwich at lunch or the bread on the table at dinner. Seems pretty straightforward, doesn't it?
Yeah, just everyday food.
But all of these things often come from just one type of plant wheat. It's kind of amazing, really. How can one single species provide so much of the world's food?
Well, it all boils down to two absolutely fundamental and really intricate processes in the plant's metabolism, photosynthesis and respiration.
Right.
So this deep dive is our chance to explore how these two processes work together. They don't just power wheat, they power pretty much all life on Earth. We're talking about the journey from sunlight all the way to the energy buzzing around in your own cells.
So our mission today is to really get into the weeds, you could say, on this elegant dance between photosynthesis and respiration. We want to unpack how they work, how they're connected in surprising ways, and, their massive impact on life.
And we'll be drawing on some excellent source material here from Plant Biology 2nd edition to guide us through it.
Fantastic. So let's start at the top. Life on Earth.
It's basically solar powered, right? It all comes back to the sun.
That's the fundamental concept, exactly. Photosynthesis is that first crucial step where cells, like in a wheat plant, they literally trap sunlight energy.
Trap it how?
They convert that light energy into chemical energy, storing it in the bonds of simple sugars. And from those basic sugars, the plant then builds everything else it needs, all the complex carbohydrates, fats, proteins, even DNA, all the building blocks of life.
Okay, so photosynthesis builds things up using sunlight, and then respiration. That's the other side of the coin.
Precisely. Respiration is the set of chemical reactions that sort of unlocks or releases the energy stored in those organic molecules that photosynthesis created.
And there are different ways to do that, right? Different types of respiration.
That's right. Two main forms. You've got aerobic respiration, which happens when oxygen is around. That's the really efficient one where sugars are broken down completely into carbon dioxide and water, releasing a lot of energy.
But then, what if there's no oxygen? Well, cells like yeast can still get some energy out. They convert sugars into alcohol instead. We call that fermentation.
Fermentation. So that's like bread and beer.
Exactly. It's the process behind bread rising the CO2, makes the bubbles, cheese production, brewing, winemaking, all based on fermentation when oxygen is scarce.
Fascinating. So let's loop back to wheat then. You said it relies on both. How did wheat even become such a staple?
Well, its story starts in the Near East, maybe over 9,000 years ago. Wild wheat actually hybridized, it sort of crossed with another wild grass, goat grass actually.
Really?
Yeah, that gave rise to emmer wheat. That's the kind often used for pasta. Then, maybe 8,000 years ago, emmer wheat hybridized again with goat grass. And that gave us bread wheat. The key difference with bread wheat is it has more gluten. That makes the dough stretchy, allows it to trap the CO2 from yeast fermentation, and lets leavened bread rise properly. For many cultures, bread became, and still is, a huge part of their diet, sometimes more than half.
And underlying all of that, the growth of the wheat, the rising of the bread, is this constant back and forth between photosynthesis capturing energy and respiration releasing it.
Absolutely. And if you zoom out even further, look at the whole planet, the biosphere, that thin skin of life. It's almost entirely powered by photosynthesis. It's the main route for energy to enter the living world.
Taking simple stuff like CO2 and water.
Exactly. Low energy inorganic stuff. And using sunlight to build high energy organic compounds like sugars, releasing oxygen as a byproduct. The basic equation, captures the what's carbon dioxide plus water with light energy, yields sugar and oxygen.
And respiration then runs that equation essentially in reverse.
Right.
Sugar plus oxygen yields carbon dioxide, water, and crucially, energy.
Right. Now, you could just burn sugar. Sure, you'd release energy, but mostly as heat. Respiration is much smarter. It releases the energy gradually and captures a big chunk of it, maybe around 38% in the form of ATP.
ATP, that's the cell's energy currency, right?
That's the one, adenosine triphosphate. It's the molecule cells use to power almost everything they do.
So how does the cell manage these incredibly complex chemical transformations? There must be some tools involved.
Oh, absolutely. The cellular toolkit is key. First, you have enzymes. Think of them as biological catalysts. They speed up reactions incredibly, millions or billions of times faster without getting used up themselves.
Okay.
Then you have the nature of the reactions themselves. Some reactions release energy. Those are called exergonic, like respiration breaking down sugar. Others need energy put into habit. Those are endergonic, like photosynthesis building sugar.
And cells link these together somehow.
Exactly. That's called coupled reactions. The cell uses the energy released from an exergonic reaction to drive an endergonic one. And ATP is often the middleman, the energy carrier. It has 3 phosphate groups, and breaking off that third phosphate releases a nice packet of usable energy.
So ATP carries the energy, but how does the energy actually move between molecules in these reactions? Is it like passing a hot potato?
It's kind of like that, but with electrons. That's where oxidation-reduction reactions come in. We often call them redox reactions for short.
Redox.
Okay, it's all about electron transfer. Oxidation is losing electrons.
Right.
Reduction is gaining electrons. Remember, oil rig. Oxidation is loss, reduction is gain.
Ah, okay, I remember that.
And they always happen together in pairs. One molecule gets oxidized, another gets reduced. Often, a hydrogen atom, which is just a proton and an electron, moves along with the electron.
Can you give an example?
Sure. In respiration, glucose gets oxidized. It loses electrons and hydrogens. Oxygen gets reduced. It gains electrons and hydrogens, forming water. In photosynthesis, it's the reverse. Water gets oxidized, losing electrons, and carbon dioxide gets reduced, gaining them to become sugar.
And these electrons don't just jump randomly.
Not at all. They are passed along specific pathways called electron transport chains. Imagine a series of molecules lined up, each one slightly more attracted to electrons than the last. Electrons get passed down the line, like a tiny bucket brigade. And as they move down this chain, they lose a bit of energy at each step. The cell captures that released energy and uses it to do work like pumping protons or making ATP. These chains are absolutely fundamental to both photosynthesis and respiration.
So it's not just random chemistry. It's organized into these pathways, sometimes linear, sometimes cycles.
Exactly. Highly organized sequences of reactions. And yes, both photosynthesis and respiration feature crucial cyclic pathways within their overall process.
Okay, this is intricate stuff, but you mentioned photosynthesis changed the planet. Here's where it gets really interesting, right? How did it literally reshape Earth's history?
Oh, profoundly. I mean, picture the early Earth. The atmosphere was totally different. Thick with CO2, methane, other gases, but almost zero oxygen. Completely alien compared to today.
Hard to imagine.
Then, maybe 3.5 billion years ago, these tiny photosynthetic microorganisms show up, probably in the oceans. They start using sunlight, consuming CO2, and releasing oxygen. Just as a waste product initially.
Well, the waste product that changed everything.
Everything. It took a long time, maybe billions of years, for that oxygen to slowly build up in the atmosphere. But as it did, something amazing happened. It formed the ozone layer, O3, high up.
The sunscreen layer?
Our planet's natural sunscreen, exactly. It started shielding the surface from the sun's harshest UV radiation. And that was a game changer. It likely paved the way for the explosion of more complex multicellular life in the oceans around 600 million years ago. And then, only after that protection was in place could life begin to colonize land, maybe 450 million years ago.
Wow. So our existence, walking around on land, breathing oxygen, we owe it all to those ancient microbes.
Fundamentally, yes. It's quite humbling when you think about it.
And what about today? What's the scale of photosynthesis now?
It's staggering. Photosynthetic organisms churn out over 250 billion metric tons of carbohydrates every single year.
Billion, with a B.
With a B. That massive production supports millions of species. For humans, just think about cereal crops, wheat, rice, corn, barley. They alone make up something like 60% of all the calories humans consume annually.
That's incredible. But it's not just food, is it?
Oh, not by a long shot. Think about materials, cotton for your t-shirt, linen sheets, photosynthesis, wood for building houses, making paper, photosynthesis, spices, perfumes, many drugs, cosmetics, waxes, oils, rubber. The list goes on and on.
And energy, too, right? Not just the food energy.
Absolutely. Many people still rely on wood-stored solar energy for cooking and heating.
Yeah.
And then there are the fossil fuels, coal, oil, natural gas. What are they? They're essentially fossilized photosynthesis, solar energy captured by organisms millions of years ago.
Which brings up a critical point. Those are non-renewable. We're burning through millions of years of stored sunlight very quickly.
Precisely. And burning them since the Industrial Revolution has pumped a lot of that ancient carbon back into the atmosphere as CO2.
Mm-hmm.
We've seen atmospheric CO2 go from around 0.028% pre-industry up to about 0.037% now, maybe higher. And that's, as we know, a major driver of global warming.
Is there any sort of counter effect from current photosynthesis?
Well, what's fascinating is that increased photosynthesis has acted as a partial buffer. Plants and oceans have absorbed a significant chunk of that excess CO2, storing it away in biomass, soils, peatlands, and ocean water. Without that, the atmospheric increase would be even worse.
It's a dynamic system. So let's get back to the mechanism. How do plants actually capture that sunlight? You mentioned pigments.
Right. They use specific molecules called pigments. And photosynthesis primarily uses visible light, that rainbow spectrum we can see. It's just a tiny slice of the whole electromagnetic spectrum.
Different light has different energy, doesn't it?
It does. Longer wavelengths, like radio waves or infrared, have lower energy. Shorter wavelengths, like UV or X-rays, have much higher energy enough to break chemical bonds, which can be damaging. Visible light is kind of in the sweet spot.
The Goldilocks zone for energy?
Sort of. It has enough energy to excite electrons in organic molecules, but generally not enough to break the molecules apart. When a pigment molecule absorbs a photon of light, one of its electrons jumps from its normal energy level, its ground state, to a higher energy level, an excited state.
And what happens to that excited electron?
It's unstable. That energy has to go somewhere. It could be released as heat or maybe a little flash of light fluorescence, or the energy could be passed to a neighboring molecule. Or, crucially for photosynthesis, the excited electron itself can be transferred to another molecule, an electron acceptor. That's the key step in converting light energy to chemical energy.
And the main pigment doing this is chlorophyll.
Chlorophyll A, yes, that's the most abundant one. It absorbs mainly red and blue violet light and reflects green light, which is why plants look green to us. But there are also accessory pigments, like chlorophyll B and various carotenoids, the pigments that make carrots orange and leaves yellow in the fall. They absorb wavelengths that chlorophyll misses and pass that energy to chlorophyll A, broadening the range of light the plant can use.
Clever. And where does all this pigment action happen inside the plant cell?
Inside specialized organelles called chloroplasts. You find loads of these, primarily in the mesophyll cells, the sort of middle layer inside leaves.
And chloroplasts have their own internal structure?
They do. Inside, there's a complex system of interconnected membranous sacs called thylakoids. Often these are stacked up like pancakes into structures called grana. The thylakoid membranes are where the pigments are located and where the light-dependent reactions happen.
Okay, membranes.
And surrounding the thylakoids is a dense fluid called the stroma. That's where the next stage, the carbon fixation reactions, takes place. Leaves themselves are beautifully adapted for this whole process: large surface area to catch sun, veins to deliver water, and tiny pores called stomata underneath to let CO2 in.
Okay, so let's break down photosynthesis into its two main stages. You mentioned the light-dependent reactions first, in the thylakoids.
Right, the light reactions. This is where the photo part happens.
Yeah.
Light energy absorbed by chlorophyll and accessory pigments gets things going. It excites electrons. This captured energy does three main jobs.
Three jobs.
Yeah. One, it powers the splitting of water molecules. That's called photolysis. This releases oxygen gas, the oxygen we breathe, plus electrons and protons, which are hydrogen ions.
Okay, splitting water, releasing oxygen. What else?
Two, some of that light energy is used directly to make ATP, that energy currency molecule.
Got it. ATP production.
And three, the energized electrons, along with protons from the water splitting, are transferred to an electron carrier molecule called NADP plus N, reducing it to form NADPH. NADPH is like another energy shuttle molecule carrying high-energy electrons.
So light splits water, makes ATP, and makes NADPH. How does the electron flow work? You mentioned chains before.
Exactly. It involves two main clusters of pigments and proteins called photosystem 2 and photosystem 1. Think of them as working in sequence. Light hits photosystem 2, exciting electrons taken from water. These electrons are passed down an electron transport chain.
Like the bucket brigade again.
It's just like that. As they move down the chain, their energy is used to pump protons from the stroma into the thylakoid space, the lumen inside the thylakoid sacs.
Building up a proton concentration.
Right. Then the electrons reach photosystem I, get another boost of energy from light, and are passed down another short electron transport chain to ultimately form NADPH. This whole zigzagging energy path of the electrons is sometimes called the Z scheme.
So we've got NADPH, some ATP, and a buildup of protons inside the thylakoid. What happens with those protons? That sounds like stored energy.
It absolutely is. That proton gradient across the thylakoid membrane, lots of protons inside, fewer outside in the stroma, is like a charged battery. It represents potential energy. This brings us to chemiosmosis and photophosphorylation.
Okay, lay it on me.
There's a remarkable enzyme complex embedded in the thylakoid membrane called ATP synthase. It acts like a tiny revolving door or turbine. It provides a channel for those protons to flow back out down their concentration gradient from the lumen back into the stroma.
Following the gradient, releasing energy.
Exactly. And as the protons flow through ATP synthase, the enzyme uses that flow energy, that's the chemiosmosis part, to stick a phosphate group onto ADP, making ATP. Because the initial energy came from light, We call this whole ATP synthesis process photophosphorylation.
Light-powered phosphorylation makes sense. So the light reactions gave us ATP and NADPH using light energy in water and releasing oxygen. What's next?
Next up is the second act. The carbon fixation reactions, better known as the Calvin cycle. This all happens out in the stroma, the fluid surrounding the thylakoids.
Okay. And this uses the ATP and NADPH.
Precisely. The Calvin cycle takes the chemical energy stored in ATP and NADPH from the light reactions and uses it to take carbon dioxide from the air and fix it or incorporate it into organic molecules. Basically, it builds sugar.
How does it grab the CO2?
It uses a crucial enzyme called rubisco, ribulose 1-corpyl-5-bisphosphate carboxylase oxygenase. Quite a mouthful.
Rubisco, I've heard that's like the most abundant protein on Earth.
It very well might be. It's incredibly important. Rubisco takes a molecule of CO2 and attaches it to a 5-carbon sugar called RuBP, ribulose 1,4,5-bisphosphate. This immediately creates an unstable 6-carbon molecule that splits into 2 3-carbon molecules.
Ah, so that's why it's sometimes called the C3 pathway, because the first stable product has 3 carbons.
Exactly right. And this C3 pathway is used by the vast majority of plants, something like 85%, including wheat, rice, potatoes, most trees. The cycle then uses ATP and NADPH in a series of steps to regenerate the starting RuBP molecule while also producing a small 3-carbon sugar phosphate called PGAL or G3P.
So the actual product isn't glucose right away.
Not directly. 2 of these 3-carbon PGAL molecules can be combined to make a 6-carbon sugar, like glucose or fructose. The plant can then use these sugars immediately for energy via respiration, convert them into sucrose, table sugar, for transport to other parts of the plant like roots or fruits, or link them together to form starch for long-term storage, maybe in the roots or seeds.
Okay, that makes sense. But I remember reading that rubisco isn't perfect. It can make a mistake.
Ah, yes, the Achilles heel of C3 plants, photorespiration. Rubisco evolved, remember, when atmospheric oxygen levels were very low, so it's not perfectly specific for CO2. It can also bind with oxygen.
What happens then?
Well, under certain conditions, especially when it's hot and dry, plants close their stomata, those leaf pores, to save water.
Makes sense.
But this also means CO2 levels inside the leaf drop and oxygen levels rise, because photosynthesis is still splitting water. When CO2 gets low and oxygen is high, rubisco starts grabbing oxygen instead of CO2.
And that's that.
It's wasteful. It triggers a process called photorespiration that actually consumes oxygen, uses ATP and NADPH, and releases already fixed carbon back as CO2. It basically undoes some of the work of photosynthesis, reducing the plant's overall efficiency, sometimes significantly.
Ouch. So evolution must have come up with some solutions, right? You hear about C4 and CAM plants.
Indeed. Nature is clever. C4 and CAM plants represent 2 major evolutionary workarounds for this photorespiration problem, especially in hot climates.
Okay, tell me about C4 plants first, like corn or sugarcane.
Exactly. Corn, sugarcane, sorghum, many tropical grasses, including crabgrass, they use a strategy of spatial separation. They have a special leaf anatomy. In their outer mesophyll cells, they use a different enzyme, PEP carboxylase, to initially fix CO2.
Not rubisco?
Nope. PPP carboxylase is much better at grabbing CO2, even at low concentrations, and it doesn't bind with oxygen. It attaches CO2 to a 3-carbon molecule to form a 4-carbon compound, hence the name C4. This 4-carbon compound is then transported into specialized inner cells called bundle sheath cells, which surround the leaf veins. Inside these deep cells, the 4-carbon compound releases the CO2. This builds up a very high concentration of CO2 right where rubisco and the Calvin cycle are located.
Ah, so it's like they pump CO2 directly to rubisco, keeping it away from oxygen.
That's a great way to think about it. It effectively swamps rubisco with CO2, minimizing photorespiration. This makes C4 plants super efficient photosynthesizers in hot, bright conditions. The downside is that this CO2 pumping mechanism costs a bit more ATP energy than the standard C3 pathway.
So C4 is spatial separation. What about CAM plants, like cacti and succulents, your snake plant example earlier?
Right. CAM stands for crassulacean acid metabolism, first studied in jade plants, Crassulaceae. These plants, typically found in deserts or very dry environments like cacti, agaves, pineapples, and yes, snake plants, use temporal separation, time separation.
How does that work?
They do something really cool. They only open their stomata at night when it's cooler and more humid, reducing water loss. During the night, they take in CO2 and fix it using that same C4 pathway, PEP, carboxylase, into 4 carbon organic acids, mainly malic acid, which they store in their cell vacuoles.
Storing CO2 overnight.
Storing it as an acid, yes. Then, during a hot, dry day, they keep their stomata tightly closed to conserve water. But inside the leaf cells, they release the CO2 from the stored malic acid. This provides a concentrated supply of CO2 for Rubisco and the Calvin cycle to use, powered by the sunlight available during the day.
Wow, so they separate the initial CO2 capture at night from the actual sugar building during the day.
Precisely. It's a fantastic adaptation for extreme dryness, but it generally results in slower growth compared to C3 or C4 plants because they can only store so much acid overnight, and they often don't tolerate cold well.
It's really incredible, the diversity of strategies, C3, C4, CM, all optimizing photosynthesis for different environments.
It truly showcases the power of evolution in adapting fundamental processes to different ecological challenges.
Okay, so plants have built these sugars, these amazing energy stores, using sunlight. Now, how does everything else, including the plant itself, actually use that energy? We need to talk about respiration.
Right. Because making sugar is only half the story. You need to be able to access that stored energy when and where it's needed. And that's the job of cellular respiration. It happens in all living cells, plants, animals, fungi, microbes.
Including the plant cells that just did photosynthesis.
Absolutely. Plant cells respire too, breaking down some of the sugars they just made to power their own growth, repair, and maintenance. Think of it like an athlete, carb loading, storing energy, and then using that energy during the race. Plants store energy via photosynthesis and release it via respiration.
So what's the basic definition of respiration then?
Cellular respiration is the metabolic process where glucose usually is broken down, typically using oxygen, to release the stored chemical energy. And crucially, a significant portion of that energy is captured in the form of ATP.
How efficient is it? You mentioned ATP yield earlier.
It's remarkably efficient for a biological process. Breaking down one molecule of glucose through aerobic respiration can yield optimally up to 36 or 38 ATP molecules. That's capturing about 38% of the energy that was stored in the glucose bonds. The rest is released as heat, which is also useful for warm-blooded animals, of course.
Okay, 38 ATP sounds like a lot. How does the cell manage this breakdown in energy capture? You said 5 main steps.
Yes, it's a multi-stage process,
Right.
each step controlled by specific enzymes. We could break it down into glycolysis, then acetyl coenzyme A formation, the Krebs cycle, the electron transport chain, and finally chemiosmosis and oxidative phosphorylation.
A lot of steps. Where do these happen in the cell? You mentioned mitochondria earlier.
Right, mitochondria are the powerhouses.
Yeah.
But the very first step, glycolysis, actually happens out in the cytoplasm, the main fluid of the cell. The remaining 4 stages all occur inside the mitochondrion. Acetyl-CoA formation and the Krebs cycle happen in the inner fluid-filled space, the mitochondrial matrix. And the electron transport chain and oxidative phosphorylation machinery are located on the highly folded inner mitochondrial membrane. Those folds are called cristae.
Those folds increase the surface area, right? More space for the machinery.
Exactly. More surface area means more ATP production capacity.
Okay, let's walk through it. Step 1, glycolysis in the cytoplasm. What happens there?
Glycolysis literally means sugar splitting. It takes 1 molecule of glucose, which is a 6-carbon sugar, and breaks it in half into 2 molecules of the 3-carbon compound called pyruvate.
Splitting glucose into 2 pyruvates, does it need oxygen?
Nope, that's
Yeah.
key. Glycolysis is anaerobic. It does not require oxygen. And it actually generates a small amount of energy directly, a net gain of 2 ATP molecules and also 2 molecules of the electron carrier, NADH.
So a little bit of ATP and some NADH, even without oxygen. What happens next to pyruvate if there's no oxygen? You mentioned fermentation before.
Right. If oxygen isn't available, the cell needs a way to regenerate the NAD plus needed to keep glycolysis going, because glycolysis uses NAD plus to make NADH.
Right.
Fermentation pathways do just that without producing any more ATP.
So it's just about keeping glycolysis running.
Essentially, yes,
Yeah.
to get that small yield of 2 ATP per glucose. In alcohol fermentation, which yeasts and many plant cells can do, pyruvate is converted into ethyl alcohol and carbon dioxide. The CO2 makes bread rise. The alcohol is obvious in brewing. If oxygen is present, pyruvate takes a different path. It gets actively transported from the cytoplasm into the mitochondrial matrix. This leads to step 2, acetyl coenzyme A formation.
Okay, inside the mitochondria now.
Right. Inside the matrix, each 3-carbon pyruvate molecule undergoes a conversion. 1 carbon atom is removed and released as carbon dioxide. This is some of the CO2 we breathe out. The remaining 2-carbon unit, called an acetyl group, is then attached to a carrier molecule called coenzyme A. The result is acetyl-CoA. This step also produces another molecule of NADH per pyruvate.
So pyruvate becomes acetyl-CoA, releasing CO2 and making NADH. Got it. What's next? Step 3.
Step 3 is the famous Krebs cycle. also known as the citric acid cycle. This also happens in the mitochondrial matrix.
Okay. What does acetyl-CoA do here?
The 2-carbon acetyl group from acetyl-CoA enters the cycle by combining with a 4-carbon molecule, oxaloacetate, to form a 6-carbon molecule, citrate, hence the name citric acid cycle. Then through a series of enzyme-catalyzed steps, the cycle systematically breaks down that acetyl group. The 2 carbons from the acetyl group are released as 2 molecules of carbon dioxide. The 4-carbon oxaloacetate molecule is regenerated at the end, ready to accept another acetyl group. So it truly is a cycle.
And what about energy capture during the cycle?
For each turn of the cycle, processing one acetyl group, the energy released is captured as 1 molecule of ATP, 3 molecules of NADH, and 1 molecule of another electron carrier called FADH2.
Okay, so per glucose molecule, which gave us 2 pyruvates and thus 2 acetyl-CoAs.
The Krebs cycle runs twice. So from 1 starting glucose, the Krebs cycle yields a total of 2 ATP, 6 NADH, and 2 FADH2.
So after glycolysis in the Krebs cycle, we have a little bit of ATP directly, but a whole lot of energy stored in NADH and FADH2.
Exactly. You can think of NADH and FADH2 as carrying high-energy electrons. They're like loaded delivery trucks carrying potential energy, or like the crude oil you mentioned earlier. Most of the energy originally in glucose is now held in these electron carriers, waiting to be converted into the refined fuel of ATP.
Which brings us to the final stages. The electron transport chain and oxidative phosphorylation, the big payoff.
This is definitely the big payoff stage. Step 4 is the electron transport chain, ETC. This is that series of protein complexes we talked about embedded in the inner mitochondrial membrane, the cristae.
Right.
The NADH and FADH2 molecules produced earlier now arrive at the ETC and drop off their high-energy electrons. These electrons are passed down the chain from one protein complex to the next, moving to slightly lower energy levels at each step.
Like cascading down a waterfall.
That's a good analogy. And just like a waterfall can turn a turbine, the energy released as electrons move down the ETC is used by some of the protein complexes to actively pump protons, H+ ions, from the mitochondrial matrix across the inner membrane into the inner membrane space, the space between the inner and outer mitochondrial membranes.
Pumping protons out, creating another gradient, just like in photosynthesis, but in a different place.
Exactly the same principle, building up a high concentration of protons in the intermembrane space. Now, what's absolutely critical here is the final step of the ETC. What happens to the electrons once they reach the end of the chain?
They have to go somewhere, right?
They do. And this is where oxygen comes in. Oxygen is the final electron acceptor. It picks up the electrons at the end of the chain and also combines with protons from the matrix to form water, H2O. This is why we need to breathe oxygen to keep the electron transport chain clear and running.
So if there's no oxygen?
The chain backs up, electrons can't be passed along, proton pumping stops, and ATP synthesis grinds to a halt. That's why things like cyanide, which block the final step of electron transfer to oxygen, are so deadly they shut down aerobic energy production completely.
Wow. Okay, so the ETC uses electron energy to pump protons, creating a gradient, and oxygen clears the electrons at the end. Now for the ATP synthesis, step 5.
Step 5, chemiosmosis and oxidative phosphorylation. That proton gradient built up by the ETC across the inner membrane now represents stored potential energy and electrochemical gradient, sometimes called the proton motive force.
The charged battery again?
Exactly. And just like in the chloroplast, the mitochondrion has the ATP synthase enzyme complex embedded in its inner membrane. This enzyme provides a channel for the protons to flow back down their gradient from the high concentration in the inner membrane space back into the matrix.
Chemiosmosis.
That's the chemiosmosis part, the flow of ions across the membrane. And as the protons rush back through ATP synthase, the enzyme harnesses that flow energy to drive the synthesis of a large amount of ATP from ADP and phosphate.
And this is oxidative phosphorylation.
Yes, because the whole process relies on the electron transport chain, which requires oxygen as the final electron acceptor, oxidative, and it results in the phosphorylation of ADP to ATP. This stage is where the vast majority of ATP from respiration is generated, potentially up to 34 ATP molecules per glucose.
So adding that to the few from glycolysis and the Krebs cycle gives us that total of potentially 38 ATP.
That's the theoretical maximum, yes. The actual yield can vary a bit depending on cellular conditions, but it's vastly more energy than the 2 ATP gained from glycolysis alone during fermentation.
It's truly an intricate and incredibly productive process. When you put it all together, Photosynthesis and respiration.
It's really quite elegant, isn't it? You see this incredible symmetry. Photosynthesis uses light energy to generate ATP and NADPH, which then power the building of sugars, storing energy. Respiration carefully breaks those sugars down, transferring the energy via NADH and FADH2 to an electron transport chain that generates a proton gradient, which then drives the synthesis of lots of ATP.
And both rely on those electron transport chains, proton gradients across membranes, and that amazing ATP synthase machine.
Fundamentally, yes. Similar machinery, similar principles, just running in different contexts, one capturing light energy, the other releasing chemical energy. It's the core energy transaction system of life.
So, okay, stepping back, what does this all really mean for us, for the listener?
It means that the energy powering your muscles as you walk, the energy powering your brain as you think about this, the warmth of your body, it all traces back. Through the food you ate, through the plants or animals that ate plants, all the way back to sunlight captured perhaps weeks, months, or even years ago by photosynthesis.
It connects us directly to the sun through these microscopic processes.
Absolutely. The air you breathe, the oxygen, is a direct product of photosynthesis splitting water. The CO2 you exhale is a direct product of respiration breaking down your food. It's this constant planet-spanning cycle, this ceaseless microscopic dance of energy transformation. It really is a testament to life's incredible, intricate engineering.
It makes you think, though. All of complex life really hinges on these tiny, invisible components, enzymes like Rubisco doing their job correctly, electrons flowing just right across membranes.
It's a finely tuned system built over billions of years.
So what happens as we start tinkering with it? Through things like genetic engineering of crops to maybe make photosynthesis more efficient, or by continuing to change the atmospheric balance of CO2 that these systems evolved with, are we pushing these ancient processes towards new limits? How does really understanding this fundamental biochemistry, photosynthesis, and respiration change how we should approach the huge challenges we face regarding energy, food security, and climate change? Something to really mull over.